Hello friends, welcome back to another video tutorial from Arun's Biology. In today's lecture, we'll discuss about the salient features of DNA double helix. So let's begin our discussion. Watson and Crick in the year 1953 first proposed the double helical model of DNA, and it was based on the X-ray crystallographic results from Wilkins and Rosalind Franklin. So let's discuss the salient features of this model. So this is the structure of a DNA double helix. So DNA double helix is basically made up of two polynucleotide chains which are anti-parallel in nature. That means the orientation of one polynucleotide chain is from 5' to 3' direction, while the orientation of another polynucleotide chain will be from 3' to 5' direction. So these two are anti-parallel to each other. If the orientation is like this, the other polynucleotide chain will be oriented like this. So they are basically anti-parallel in nature. Coming to the second point, which is the polynucleotide chain. Each polynucleotide chain is basically made up of sugar phosphate backbone from which the nitrogenous bases are projected inward. Okay, so how this occurs? If suppose this is a polynucleotide chain, here we can draw it like this. If it is a phosphate, it is connected with a sugar, then it is connected with a phosphate, again a sugar. So this is the 5' end of this polynucleotide chain, this will be the 3' end of the polynucleotide chain. And the sugar and phosphate, that is the phosphate group and this deoxyribose sugar remain connected with each other with the help of 5', 3' phosphodiester bond. So this will be the orientation or arrangement of the sugar phosphate backbone of the other polynucleotide chain. So this is the orientation 5' to 3', and its orientation is from 3' to 5'. While the nitrogenous bases remain connected with this sugar. Okay, here also the sugar will have this nitrogenous base with the help of N-linked glycosidic bond. And this nitrogenous bases will remain joined with each other with the help of hydrogen bond in between them. So this conformation will stabilize this DNA double helix. Coming to the third point, which is complementary base pairing. So in the DNA, we'll have different purine and pyrimidine nitrogenous bases. What is the meaning of complementary? Complementary means they are not identical. Okay, so if it is a adenine, that is a purine nitrogenous base, it will bind with a thymine, that is a pyrimidine type of nitrogenous base, and they will form two hydrogen bonds in between. If suppose it is a guanine, that is again a purine nitrogenous base, it will bind with the cytosine, which is a pyrimidine nitrogenous base, with the help of three hydrogen bonds. So the hydrogen bonds are formed like between A and T there is two hydrogen bonds, and between G and C there is three hydrogen bonds. Now coming to the fifth point, which is the helical pattern of DNA, and it is basically a right-handed helix. So if we talk about the different types of DNA, there are different types from A to E. Okay, A, B, C, D, E, there are different types of DNA, and all this DNA are right-handed helix except one, which is this DNA, which is the ZDNA. Okay, ZDNA or zigzag DNA, it is basically left-handed helix, while all other DNA is right-handed in nature. Coming to the major and minor groove. So this is another important feature of a DNA double helix. It will have a major groove and minor groove. Due to this helical pattern or this twist of this DNA molecule or the two polynucleotide chains which form the DNA molecule, will have minor groove and major groove in an alternate fashion. Okay, so one minor groove is alternating with a major groove, and then again a minor groove will come. So the distance between two consecutive major groove or two consecutive minor groove is called one pitch or one complete turn of a DNA double helix. And what is the distance? If we calculate the distance of this one pitch, it will come around 3.4 nanometer, or it is equivalent to 34 angstrom. And in this 34 angstrom we'll have 10 base pair. Okay, so the distance between two consecutive base pair, if suppose this is a base pair G and C, another base pair is T and A, so the distance between these two will come like 34 angstrom divided by 10, so it will come 3.4 angstrom. So very simple calculations can come from this part. Okay, in the examination you can be provided with the length of a DNA fragment. If it is say for example 1.36 millimeter length of the DNA fragment, you need to calculate the number of base pair that is present in this 1.36 millimeter of DNA. So how we will solve this? We know that 3.4 angstrom means the distance between two base pair or the distance between two nitrogenous bases. So suppose this is the distance between two nitrogenous bases. So this distance is 3.4 angstrom. Okay, so 3.4 angstrom means one base pair. That means 1 angstrom distance will be covered by 1 divided by 3.4 base pair. This is a unitary method. You might have understood very easily. So if the distance is 1.36 millimeter, we will convert it in angstrom by multiplying it with 10 to the power 7, and final calculation it will come 0.4 into 10 to the power 7. That means 4 into 10 to the power 6 base pair. So we can write the answer like in 1.36 millimeter distance of or length of DNA will have 4 into 10 to the power 6 base pair. This kind of very simple numericals can come in the board exams. Okay, this question can be asked in a different way. They can give you the number of base pairs. Say for example, we take the number of base pair here like 1000. If it is a 1000 base pair, it contains 1000 base pair, what will be the length of this DNA? It is quite easy. So we will just multiply it with 3.4 angstrom. So the distance will come. Okay, and in a second question they can ask you to calculate the number of base pair from the length of the DNA given. Okay, coming to the next important salient features of this DNA double helix, that is the pitch. We have already discussed this thing. The distance between two consecutive base pair is 3.4. So these 2 points we have already covered. Now the distance between two polynucleotide chain, that is 20 angstrom. Okay, and this distance remain constant throughout this polynucleotide chain. Now how this two distance remain constant? For answering this question, okay, we need to know that what are the forces that stabilizes the DNA double helix. So there are basically two forces that stabilizes the DNA double helix, and what are these? The hydrogen bond between the complementary nitrogenous bases. So if these two are nitrogenous bases, the hydrogen bond in between will stabilize this DNA double helix. And the second is the hydrophobic interaction or the base stacking force between the nitrogenous bases. So this is a nitrogenous base, and this is again another nitrogenous base. So these bases are hydrophobic in nature, so they will interact with each other with the help of hydrophobic interaction. So the hydrogen bond between the nitrogenous bases of two different polynucleotide chain and the hydrophobic interaction between the nitrogenous bases actually stabilizes this DNA double helix, and as a result the distance between two polynucleotide chain remain constant. Okay, so this is all about the salient features of DNA double helix. So we need to memorize these salient features. These are pretty important for the examination purpose. We need to know the simple calculations, and one important question, why the distance between two polynucleotide chain remain constant. So we have to write the name of the forces that stabilizes this DNA double helix. So this is all about the salient features of a DNA double helix. So in the next topic or next video lecture, we'll discuss about the Stargaff's rule and quite important numerical problems that generally come in the board exam. Thank you for watching.